Hello students. Now continuing with the third part of the development of the third week, I'll start with the formation of neural tube. So firstly, what is neurulation? So you get a short note on the definition of the neurulation and you have to enlist various steps involved in the neurulation. So firstly, we have to understand this is a trilaminar embryonic disc. The notochord has been already formed. Now this notochord is the primary inducer or organizer for the neural plate formation. That is the overlying ectoderm or the epiblastic cells. They are organized into a plate-like structure which is known as neural plate. So what is the definition? The process of formation of the neural plate and neural folds and closure of these folds to form a neural tube is called neurulation. Now, so what is the process of neurulation? Firstly, it involves the formation of a neural plate, or it is also known as medullary plate. So at the beginning of the third week, the midline central part of the ectoderm, that is in between the primitive node and the precaudal plate. So lying above, this is the part of the ectoderm which is lying above the notochord. It thickens to form a neural plate. So the process of neural plate formation is called neural induction. Now the ectodermal cells differentiate into a thick plate of pseudostratified columnar neuroepithelial cells. So neural plate is made up of pseudostratified columnar neuroepithelial cells. Then is the step of neural groove formation. That is the elevation of the edges. This is an edge of the neural plate. So elevation of the edges of the neural plate to form the peripheral neural folds and a central depressed part which is known as neural groove. So just as you close your book, so similarly is the formation of neural groove, that is the mid part of your book. It forms the neural groove and the peripheries of your pages of the book will form the edges of the neural plate. So the elevation of the neural folds continue and starts fusion in a future cervical region and then extend in a craniocaudal direction to form the neural tube. So here you can see this is the fusion has started in the cervical region and finally it is proceeding from cranial to the caudal direction. So the conversion of the neural plate into the neural tube is called neurulation. So the cranial and the caudal neuropores are open ends of the neural tube. Since a tube is formed, so it has a cranial pore and the caudal pore. So cranial neuropore, it will close at the day 25 and the caudal neuropore will close by 27th day. Now, what are the certain neural crest cells? So this is the neural plate. You can understand this diagram. This is the notochord. So now the neural plate has been formed. Now over here, the neural groove is forming. So there, these are the two lateral ends. So the lateral lips of the neural plate give rise to an extremely important population of cells, which are known as neural crest cells, which will detach during the formation of the neural tube and they will migrate in the embryo to form a variety of structures. So during the detachment, the neural crest cells, they come to lie in between the neural tube and the surface ectoderm. So neural tube will form the central nervous system and the neural crest cells will form the peripheral and the autonomic nervous system. So neural crest cells further take two pathways. One is the dorsal pathway and another one is the ventral pathway. So prior to the closure of the neuropores, amniotic fluid circulates through this neural tube to provide the nutrition to the developing brain. So I told you that the neural crest cells, they have developed from the lateral folds. They will detach before the neural tube will fuse and they will come to lie in a position just below the ectoderm and between the neural neural tube. So they will take the two pathways, the dorsal pathway and the ventral pathway. Through the dorsal pathway, some of the neural crest cells, they will enter the ectoderm through the holes in the basal lamina to form skin melanocytes and hair follicles, right? So via dorsal pathway, the neural crest cells will form melanocytes and the skin hair follicles. So these two are also the derivatives of neural crest cells. Then some of these will take a ventral pathway through the anterior half of the each somite to become a sensory ganglia, a sympathetic ganglia, and an enteric neurons, the Schwann cells, which will insulate the peripheral nerves, and cells of the adrenal medulla. So this one is the enteric ganglia, the preganglionic or other sympathetic ganglias. These are the sympathetic ganglias. You can see the dorsal root ganglia and the developing suprarenal gland, so the adrenal medulla. So in brief, the process of neurulation, it involves thickening of the middle part of the central part of the ectoderm. It forms the neural plate. Then it forms the neural folds and neural groove. Then there will be fusion of the neural folds. Then it will lead to formation of neural tube whose cranial neuropore will close at the day 25 and the caudal one will close at day 27. Then before the neural tube is formed, there is detachment of the lateral margins of the neural folds. These are known as neural crest cells. So this is a summary that how the neurulation has been occurred. So this is a cut section at the cranial part of the notochord. So this one is the primitive streak. This one is the neural plate. Now the primitive streak with the formation of the neural plate, this is the underlying notochordal process which will induce the surface ectoderm to form the neural plate, which is expanded cranially. And similarly, you can see the primitive streak, it is regressing in the caudal area. So now you can see a slipper shaped neural plate is formed, which is lying above the notochordal process. Then what happens? How the neural tube is formed? This neural plate which is formed, it gets detached from the rest of the epiblast cells and it will start folding on each other. So this is the cut section now. So in a cross-sectional view, you can see this one is the neural plate which is induced by this notochord. Then the neural folds which are formed, this one is the central part is the neural groove. This will further deepen. These are the neural crest populations which are present on the lateral margin of this neural fold. So this neural fold, it will start fuse with each other. Before they will fuse, these neural crest cells population, they come to lie between the surface ectoderm and the neural tube. Now they will take two pathways, the dorsal and the ventral pathways. So the dorsal pathway will form the melanocytes, while the ventral pathways will form the dorsal root ganglia, autonomic ganglia, the adrenal medulla, and the enteric nerve cells. So what are the different neural crest derivatives? These are the connective tissue and bones of the face and skull, cranial nerve ganglia, cells of the thyroid gland, that is the parafollicular C cells, the conotruncal septum in the heart, the odontoblasts of the teeth, dermis of face and neck, spinal dorsal root ganglia, sympathetic chain and the preaortic ganglia, the parasympathetic ganglia of the gastrointestinal tract, adrenal medulla, the Schwann cells, the glial cells, meninges, melanocytes, and the smooth muscle cells to blood vessels of the face and forebrain. So these all are derivatives of the neural crest cells. So you get a short note on the neural crest cells. So you have to write what are the events which lead to the formation, how there are two pathways, and what are their derivatives. Now there are certain NTDs or the neural tube defects. So when the neural tube, it fails to close, then the certain defects occur. So these are known as neural tube defects. If the neural tube fails to close in the cranial region, then most of the brain fails to form, and the defect is known as anencephaly, as it is depicted over here. If it fails to close anywhere from the cervical region caudally, then the defect is known as spina bifida, and this defect of lumbosacral region is the most important defect. So we have a model over here. So we'll shortly post a video of all the embryo models of the related to your embryology. So that completes our third week. Thank you for listening. Any comments, you can share it over here. Thank you so much.